Good evening, everyone. Today I'll be discussing the contents of week 10, which is with regards to the nervous system, followed by some questions pertaining to that. Let me just share my screen. Um, Till now we have understood different aspects of a living organism, which pertains to how living organisms are classified, how they have evolved from the and from their ancestors, how the basic uh, thing that makes up the organisms, which is the cells, are made up, are designed, what are the molecules that make up the cell, what are the cellular processes that are affected within uh, these systems. Now we are looking at the physiology of what goes on in a human system. So, just sorry. Um, there are multiple organ systems in place to ensure the smooth functioning of any organism. Uh, in case of in lower organisms, some of these systems might be merged together. Some of these might have organ systems that repeat. But in case of humans, all of these systems are very nicely separated to ensure proper function. So, we have looked at digestion, we have looked at circulatory system and muscular system. We have we will be looking now at the nervous system, which ensures that whatever information needs to travel from one position to another is done effectively. So the nervous system coordinates physiological functions in humans. So when zygote is formed and it starts to divide, it's or at one particular stage in the developmental regime, it arranges itself into into different layers. These layers are called endoderm, ectoderm, and mesoderm. And all of these layers have specific functions. They give rise to specific organs, and this is conserved inside a species. If something gives rise to, say, the nervous system, all in every organism in that species or in their ancestral species, that particular layer is going to give rise to the nervous system. So, in case here, the nervous system originates from the ectoderm. The it is the the particular function that nervous system is responsible for is receiving stimulants and transmitting messages. So, the origin of human nervous system is ectodermal, which implies that it originates from the ectoderm layer. The whole nervous system is divided into three parts: central nervous system, peripheral nervous system, and autonomic nervous system. Central nervous system itself constitutes of brain and spinal cord. Uh, just one second. peripheral nervous system comprises of different nerves that are present throughout the body, which is the cranial nerves and the spinal nerves. The cranial nerves are essentially nerves that are present inside the head, and the spinal nerves originate from the spinal cord. And the autonomic nervous system comprises of sympathetic and parasympathetic nervous systems. So, let's go through each of these. The central nervous system, which comprises of brain and spinal cord. The brain is present inside a bony hollow known as the skull, whereas the spinal cord is protected by the vertebral column. This is important because both of these systems are very essential for functionality of a human being. But if any damage occurs to either brain or spinal cord, a lot of problems might happen depending on where the damage has happened. And both of these organs are composed of very soft tissues, because of which they don't have much. If there is a direct hit done to any of these organ systems, it is going to be very disastrous. So both of these are protected by bony elements, which is skull for the brain, and for spinal cord it's the vertebral column. Apart from the skull, the brain is also protected by a triple membrane of connective tissue known as meninges. And there are three layers to the meninges. There is dura mater, arachnoid, and pia mater. The pia mater is the innermost layer, whereas the dura mater is the outermost layer. The innermost layer is very thin, it's kind of delicate, and it's highly vascular, which implies that all of the blood flow that is going on in the pia mater. The middle layer is known as the arachnoid, which is thin and highly folded structure in front of the cranial venous sinus. The function of the arachnoid membrane is to maintain the cerebrospinal fluid's homeostasis. This fluid gives some cushioning. It is also very important that there is the volume of this fluid stays consistent. The outermost layer is very thick, strong, and non-elastic, called as dura mater. It is mostly made up of collagen fibers, which is a component of the extracellular matrix. The space between dura mater and arachnoid is known as subdural space, which is filled by serous fluid. And the space between arachnoid and pia mater is known as subarachnoid space and is filled with cerebrospinal fluid. The cerebrospinal fluid is lymph-like, clear, alkaline, whose function is to provide support as well as to protect the brain. There is a lot of homeostasis that goes into the maintaining the cerebrospinal fluid. It also helps in maintaining the metabolic status of the brain and the blood capillaries that surround the brain. So it ensures that there is no buildup of any metabolic substances inside the brain. So it is very important for the maintenance of the environment of the brain. CSF is mainly present in the ventricle of the brain, subarachnoid space, and in the spinal cord. Therefore, cerebrospinal fluid. The human brain itself has a lot of structure to it. It is divided into mainly three parts. and then these three parts contain different kinds of structures to them. Forebrain or proencephalon, midbrain or mesencephalon, hindbrain or rhombencephalon. The forebrain includes cerebrum, diencephalon, and olfactory lobes. Midbrain consists of corpora quadragemina and crura cerebri, and the hindbrain consists of cerebellum, pons varoli, and medulla oblongata. The hindbrain is also forms the connecting link between the spinal cord and the brain. So let's go through different parts of the forebrain. The cerebrum is the largest and most advanced part of the brain, which composed of two cerebral hemispheres, left and right, on the dorsal surface. So there is a groove, there is a separation between left and right hemispheres, known as the median fissure. These are connected with a curved thick nerve fibers, which are known as corpus callosum. The outer portion of the cerebrum is known as cerebral cortex, which is made up of numerous cell bodies and relatively few myelinated neurons, because there is not much need of signal to travel through a long distance. Whatever information is generated here is then transferred to the spinal cord, and which then conveys the information. So myelinated versus non-myelinated neurons come into play when there is message transfer required. So myelinated neurons mean that the neuron body is covered with myelin sheath, which prevents the membrane from the polarity of the membrane from changing. So myelin sheath kind of acts like an insulation, which prevents any signal to leak out of the nerves itself. The neurons give an overall gray appearance and hence are called as gray matter. The surface of the cortex is highly folded. The upward folds are called gyri, consecutive with downward grooves are called sulci. So these folds that are present in human brain are not present in some certain lower animals. This is especially important in case of human brain, because it increases the surface area available for information exchange, for information storage, and this also makes sure that higher level processing, cognition, consciousness can happen in case of humans. So one of the reasons that these properties are present in humans are because of the gyri and sulci kind of arrangement present in the human brain. Beneath the gray matter, there are millions of myelinated axon tracts that contain relatively few cell bodies, which give an opaque white appearance, and this is known as the white matter. So there is gray matter, there is white matter, which forms one kind of division inside the brain. In the brain itself can be divided into multiple lobes: frontal or anterior lobe, parietal or middle lobe, temporal or lateral lobe, and occipital or posterior lobe. And each of these lobes have a specific function. The central sulcus, or the median fissure separates frontal lobes from parietal lobes, and the central sulcus separates frontal lobes from parietal lobes. The lateral sulcus or sylvian sulcus separates temporal lobe from the frontal and parietal lobes. So the central sulcus separates the frontal and parietal lobe, and the lateral sulcus separates the temporal, frontal and parietal lobes. There is also a parieto-occipital sulcus that is responsible for separating the parietal and occipital lobes. Okay. So the brain looks something like this. There are these ridges which are known as gyri. There are these grooves which are known as sulcus. There are different lobes to the brain. The major portion is known as cerebrum. There is cerebellum, which is the second largest structure inside the brain. There is pons varoli, medulla oblongata, which finally connects to the spinal cord. Each of these areas are specific for a particular function. So within the cerebrum, there is a sensory area, there is an association area and a motor area. The sensory area have neurons that receive impulse from different receptors. Association area interpret the information that comes in the sensory area, store the input and initiate a response. This area is especially important for memory, learning, reasoning, making connections, etc. Motor area then transmits the impulse to effector organ. So whatever input comes in through the sensory area, the association area interprets this input, if needed stores it and initiates a response, and this response, if required, if it requires a motor response, then the motor area then transmits the impulse to the effector organs. The other parts of forebrain are diencephalon, which is a small posterior part of the forebrain, which is mostly covered by cerebrum. The diencephalon the diencephalon is kind of internal to the cerebrum. So the cerebrum covers the diencephalon. The diencephalon itself consists of thalamus, hypothalamus, epithalamus and metathalamus. Thalamus is the major part of diencephalon and represents the upper lateral wall. It accepts all sensory impulses from all part of the body except for olfaction and sends them to the cerebral cortex. Thus it acts like a relay center. In lower animals, thalamus acts as a sensory center because the cerebral cortex is less developed. So whatever information comes in from the body goes through the thalamus into the cerebral cortex. The hypothalamus is known as the master gland. It represents the lower lateral wall of the diencephalon. Pituitary gland is also attached to this with its middle part. A web-like structure is found on the anterior surface of the hypothalamus known as optic chiasma, and in mammalian brain, corpus albicans is found in posterior part of the hypothalamus. So the hypothalamus signals when different hormones should be produced. Therefore it is known as the master gland. So it has specific cells that secrete hormones that will trigger growth, which will trigger generation of different regeneration. It might trigger hunger, all of these properties. So what hypothalamus does is it produces various stimulating and inhibiting hormones, which then go to their respective endocrine glands and trigger secretion of the hormones of that specific gland. Next is epithalamus, which represents the roof of the roof of diencephalon. It can be regeneration of different cell types, especially in case of young adults or children, uh, in organ systems where regeneration is possible. So hypothalamus has multiple functions. It secretes growth hormone. Therefore can, because it secretes growth stimulating hormone, then it can trigger lengthening of bones. It can trigger muscle generation, etc. Epithalamus, did that clear the question? Was that clear? Okay, cool. Epithalamus represents the roof of diencephalon. Pineal gland is found in this region. Metathalamus represents the floor of diencephalon, and it consists of a medial geniculate body related to hearing and a lateral geniculate body related to vision. So if some portions of there is some redundancy in certain areas in the brain, but more or less every portion inside the brain has a particular function, and if there is damage to that portion, that implies that that function is going to be compromised. Next is the midbrain. It is a small contracted part of the brain. Two longitudinal myelinated nerve fibers called cerebral peduncles or crura cerebri located at the anterior part of the brain. There are four spherical projections called optic lobes or colliculus that are located in the posterior part of the midbrain, and inferior optic lobes, which are related to acoustic reflex action, are also present in the midbrain. Next is the hindbrain, which has the cerebellum, medulla oblongata, and pons varolii. Cerebellum is the second largest part of the brain after cerebrum. It is made up of three lobes. Lateral lobes are large, spherical, and called as cerebellar hemispheres. It controls regulation and coordination of voluntary muscles, and it helps in maintaining the body balance of the person. Medulla oblongata is a tubular and cylindrical in shape, present in the posterior part of the brain. It controls all the involuntary activities of the body, respiration, metabolism, secretory actions of different cells, etc. So if there is damage to medulla oblongata, the respiratory system of that person might shut down or might get depressed. Pons varolii is a small spherical projection which is situated below the midbrain and upper to the medulla oblongata. It consists of many transverse and longitudinal nerve fibers. Transverse nerve fibers are joined with cerebellum, whereas longitudinal are joined with cerebrum to join the cerebrum to the medulla oblongata. Pons varolii regulates the breathing reaction through pneumotaxic center. So if the exhale and inhale pattern of any human is controlled by pons varolii, and the midbrain, medulla oblongata and pons varolii are situated on one axis known as the brainstem. So Earlier I said that there are different lobes to the cerebrum. These lobes are responsible for different kinds of functions. Frontal lobe has different areas, so each of these lobes themselves have different areas. Frontal lobe has all of these areas, temporal lobes have all of these areas, and parietal lobes have these areas. So the first area of frontal lobe is prefrontal cortex, which is the site for intelligence, knowledge and memory. The premotor area is responsible for writing, as movement of head, eye, body, uh, complex movement of jaw, tongue, larynx, etc. And this is the area that also allows for speech to some extent, because it controls the jaw, tongue, and pharynx. Next is the motor area, which is responsible for voluntary muscles. Frontal eye field is responsible for opening and closing of eyelid and conjugate eye movement. Broca's area or motor speech area is responsible for speech along with premotor area. Temporal lobe has auditory area, olfactory area, and vernix area. Auditory area is, as the name suggests, responsible for sound. Olfactory area, smell, and vernix area is responsible for communication. So anything with olfaction in it will be responsible for smell, or if it's auditory, then it's responsible for hearing or sound. Parietal lobes has gustatory area responsible for taste, somesthetic area responsible for touch, pain, pressure, etc. Angular gyrus responsible for writing, associated with speech. Occipital area is responsible for vision. The spinal cord is, as I said, connected to the brain with via the medulla oblongata, which comes out of the foramen magnum, which is the sort of the gap in the skull where the medulla oblongata connects to the spinal cord and continues into the neural canal of the vertebral column. It is also covered by dura mater, arachnoid, and pia mater, but the thicknesses of all of these layers in the spinal cord and the brain is different. Narrow space between dura mater and vertebra is known as epidural space. The outer part of spinal cord is made up of white matter, whereas the inner part is made up of gray matter. The gray matter projects outside and forms the dorsal and ventral horn. And the dorsal ventral horn continues in a tube-like structure known as root of dorsal and ventral horn. This root has ganglia present in them known as dorsal root ganglion, and sensory neurons are found in this dorsal root ganglion, and the axons of these neurons extend and get embedded into the gray matter of the spinal cord. These sensory neurons make synapse with the ventral root neuron. So there are a lot of neurons that originate or have inputs into the spinal cord, and all of these neurons span different functions that the nervous system is responsible for. Motor neurons are found in the ventral root, whose cyton means cell body, is found in the ventral horn. where while its dendrons are embedded into the gray matter of the spinal cord. Both sensory and motor fiber are combined and comes out of intervertebral foramen and form the spinal nerve. The spinal cord essentially acts as a bridge between the brain and the organ of the body. It regulates conducts reflex action as well as provides relay path for impulses coming from the brain. Just a second. Sorry. Okay. So reflex actions are involuntary actions completed very quickly as compared to normal actions. These are different from normal actions because there is very little control to all of these actions. So they are of two types: cranial reflex or spinal reflex. The cranial reflexes are, as the name suggests, completed by the brain. These are slow actions such as watering of the mouth to see good food, etc. So salivating. Spinal reflexes are completed by spinal cord. These are fast actions such as withdrawal of the arm at the time if you touch a hot surface, or if you touch a sharp thing, then the jerking action of the arm is a spinal reflex. Okay. Next is, so this was all about the central nervous system. Next is the peripheral nervous system. In case of the peripheral nervous system comprises of cranial and spinal nerves. So all of these nerves in totality make up the peripheral nervous system. If they arise from the brain, they are cranial nerves, or if they arise from the spinal cord, they are known as spinal nerves. All of reptiles, birds, mammals, etc. have 12 pairs of cranial nerves. Lower animals like amphibians or fishes have 10 pairs of cranial nerves. And each of these nerves have a specific function in a particular species. In case of humans, for example, 1, 2 and 8 are responsible for sensory inputs. 3, 4, 6, 11 and 12 are responsible for motor nerves, whereas rest are mixed type of nerves, which can be auditory, can be responsible for vision, etc. In humans, there are 31 pairs of spinal nerves. All of these spinal nerves are mixed type, which implies they can and uh, provide receive input of multiple types and provide output of multiple types, and they arise from roots of the horns of gray matter in the spinal cord. Spinal nerves are divided into 5 groups. There is cranial spinal nerve, which is present in the head. Thoracic spinal, which has 8 pairs. Thoracic spinal nerve, 12 pairs, present in the thoracic region. 5 pairs of lumbar spinal nerve, 5 pairs of sacral spinal nerve, and 1 pair of coccygeal spinal nerve. So when you look at the vertebral column, it has thoracic, lumbar, sacral, coccygeal regions, right? So the nerves that originate in those specific regions are named in such a way. Okay. So there are 12 different cranial nerves. There is and there is particular names to them, and there is a particular function associated with it. For example, olfactory nerve arises from the olfactory epithelium. It is sensory in nature and it is responsible for smell. Optic nerve is also sensory region and is responsible for sight. There are certain nerves which are mixed in nature. So for example, ophthalmic nerve is mixed. Trigeminal nerves are mixed. They are composed of ophthalmic, maxillary, and mandibular. Facial nerves is mixed in nature. It is responsible for taste, facial expression, and saliva secretion. So each of these nerves has their own function, has their own nature, whether it's sensory or motor or both. It has a particular origin, it goes to a particular region, and this is consistent in every human. Next is the autonomic nervous system, which controls involuntary activities. For example, heart rate, sweating, peristalsis, etc. This consists of motor neurons passing to the smooth muscle of internal organs. It is also very important for maintaining homeostasis, which implies that the state of the cell or the organ system does not change much. The influx and outflux remains the same. Autonomic nervous system comprises of sympathetic and parasympathetic nervous system. The sympathetic nervous system is related with intuitive reaction, which increases the protection of the body in adverse atmospheric condition along with energy consumption. Parasympathetic nervous system is linked with reaction in which energy is conserved. So anything that consumes energy is related to the sympathetic nervous system, whereas parasympathetic nervous system has reactions which do not need energy. Okay. So nerves are very essential for the conduction of information from one place to another, and any damage to these nerves can have very deleterious effects. So it is very important in case of any accident or in case of any functional problems that the conduction of if the nerves are conducting messages properly or not is looked at. A medical diagnostic test to do that is a nerve conduction study. So it is used to estimate the function and capability of electrical conduction of both motor and sensory nerves. What is measured in the test is the nerve conduction velocity. How fast is the information how fast does the information travels from point A to point B. It can be done with an electromyography to measure muscle function as well. So NCS and electromyography will tell you about muscle and nerve function. Diagnosis of defective spinal nerve compression or any other neurologic disorder or injuries are undertaken for the study of the NCS process. Evaluation of numbness of limbs, weakness of legs, arms, as well as tingling or burning sensation in certain areas of the body at the central area of diagnosis. Nerve conduction study mainly comprises of different kinds of studies: motor NCS, sensory NCS, F-wave study, and H reflex study. So let's go one by one. Motor NCS is performed by electrical stimulation of a peripheral nerve and recording from a muscle in which these nerves are this nerve supplies. Latency is defined as the time taken for the electrical impulse to travel from the stimulation to that muscle, and is usually measured in milliseconds. So, a nerve goes from point A to point B. It is responsible for action at point B. So, the electrical impulse is applied at point A, then the time it takes for the effect to show at point B is noted down. If the latency is increased, that implies that there is something wrong with that particular nerve. Sensory NCS is similar to motor NCS, but the reaction is done from the sensory portion of the nerve. Sensory latencies are also measured in milliseconds, but the amplitudes are much smaller than motor amplitude, microvolts. Sensory velocities are also calculated as the same way as motor velocities. Next is the F-wave study, in which both the motor and sensory segments are taken into account, especially in segments where both of these are supply their both motor and sensory segments are present. It is F-wave latency is used to derive the conduction velocities of nerve between limb and spine. In a typical F-wave study, a strong electrical stimulus is applied above the distal portion of the nerve. The resulting impulse travels in both directions, one towards the muscle fiber and the other back to the motor neurons of the spinal cord. And you measure the times taken for both. You record the signal at both of these places, measure the time taken, and this will give you a idea of the latency of that nerve. H-reflex study is similar to F-wave study. It evaluates conduction between the limb and the spinal cord. There is one difference that exists, that the impulse going towards the spinal cord are in sensory nerves, while the impulse coming from the spinal cord are in motor nerves. So the interpretation of a nerve conduction study is very complex. It requires multiple practitioners such as neurologists, psychiatrists, neurophysiologists, etc. But it is very as important for diseases related to nerves. Most of the study is very non-invasive because there are electrodes placed on top of the body. There is no surgical intervention required, but there is some pain due to the electrical shocks, which is important for studying the conduction. These shocks are usually of very low amount and have been proven not to be deleterious to humans normally, but in case of people who have pacemakers, they are usually suggested not to do these studies, and alternate studies are suggested instead of these. One other test that is very important is the electroencephalogram or EEG. This is required for measuring the brain activity. So there is a cap which has multiple electrodes present at specific points placed on the human, and whatever electrical impulses are generated are recorded, are amplified and recorded. If the impulses are okay, then that implies that the brain functioning is okay. If there is any drop in the amplitude, if some of the waves are missing, then that implies that something is going wrong. It operates by detecting the brain's electrical charge, which is maintained by billions of neurons. The neurons are electrically charged due to continuous pumping of ions by membrane transport proteins across the membrane. So in the brain, in neurons, um, potassium and sodium ions play a very important role in maintaining the information transfer. If there is any problems in the gradient of the potassium or sodium ions, then that is going to either delay or lead to a very fast information transfer, which is not really essential. So this the difference in the potassium and sodium gradients is what leads to a charged system in the neurons, and this is what the EEG measures. Neurons constantly exchange ions with the extracellular fluid to maintain resting membrane potential. When many ions have similar charge that are pushed out several neurons at the same time, they can push their neighbors who further apply force to their neighbors, and so on in waveforms. When the wave of ions reaches the electrodes attached to the scalp, they give or take electrons on from the metal of the electrodes. Since metals can conduct these electrons easily, voltage difference between two electrodes can be measured by a voltmeter. Recording these voltages for a specific time gives us the EEG. There are different wave patterns that happen during EEG recording, and the EEG recording is also done at specific times. It can be done when a person just sitting, it can be done when the person is sleeping, it can be done when a person is drawing, if the person when the person is observing some a particular images which trigger something specific inside the brain. It can be done while the person is walking. There are multiple kinds of EEGs that can be done, and depending on what kind it is, it will give a different kind of graph. The wave patterns that form during EEG recording, the first one is delta waves, which is the frequency is below 4 hertz. It's highest in amplitude and it is very slow, and it is normally observed during sleep in adults, in babies as well. This even in patients who are in coma, in which case the wave patterns are very dysfunctional, even in those patients delta waves are observed. Theta waves range from 4 to 7 hertz. It is normally seen in young children or in adults or older children under stress or during medication. If there is excess of theta for a particular age, it represents abnormal activity. Alpha waves range from 7 to 14 hertz. An awake but resting person produces an alpha wave, and this posterior basic rhythm is seen in occipital regions of the brain, in the back of the brain essentially. It arises with the closing of eyes and with relaxation, and weakens with eye opening and mental labor. So the amplitude will drop according to what state the person is in. In addition to the posterior basic rhythm, there are other normal alpha rhythms such as mu rhythm, which arises when hands and arms are indolent and non-functional. Beta waves range from 15 to 30 hertz. And these are very prominent during extreme mental activity, uh, in which case they are initiated in the frontal and parietal regions. Beta activity is closely linked to the motor behavior and is generally weakened during active movements. An alert, wide awake person shows unsynchronized beta activity. Gamma waves are from 30 to 100 hertz. They represent binding of different populations of neurons together into a network for the purpose of carrying a certain motor function. Mu waves have a frequency of 8 to 13 hertz, and they are a part of alpha waves. uh, it uh, it denotes synchronous firing of motor neurons in a resting state. If there is any deviation from any of the probable patterns of these waves, that implies there is some brain or neurological disorder, and then uh, other tests can be done to figure out what exactly is wrong. EEG can diagnose epilepsy, brain tumor, abscesses, sleep disorders, metabolic and drug effects on the brain. Okay. Okay. The nervous system has neurons and neuroglia. The neurons can have like the cell body cyton, or the uh, which has most of the organelles, or the neurites, or the process of the neuron which is responsible for conduction of the information. The nervous tissue originates from ectoderm and is specialized for receiving stimuli and transmitting message. It consists of highly specialized cells known as neurons, and these are functional units of the nervous system. A neuron consists of two portions, cell body or cyton, and cell processes. This is the cell body, whereas the rest of it is known as the cell process. There is the broader part of neuron which contains uninucleated cytoplasm. Except for centriole, all types of cell organelles are found in cytoplasm, and due to absence of these centrioles, these neurons can't divide. Centrioles are very essential for formation of, for the separation of the uh, nuclear for the nuclear material into two daughter cells. Because of the absence of neurons, absence of centrioles, these neurons are not divided, and these are terminally differentiated in nature. Some other cell organelles like neurofibrin and Nissel's granules are found in the neuron, which help in transfer of impulse to the cyton. Nissel's granule is formed by the coiling of endoplasmic reticulum around ribosome. Cell process, which are dendrons and axons, are branches that originate from cell body. If the branches are even finer, in case of dendron it is known as dendrites. Some of these dendrites have receptor points, so that the dendron can receive the information that is going on, whatever stimulus is going on around here, and produces a centripetal conduction. Axon is the longest part of the neuron. So axon is a very long dendron, but the information that comes from dendron towards the axon, part where the axon arises from the cyton, is known as the cyton hillock here. The cytoplasm of axon is called axoplasm, which contains only neurofibrils and mitochondria. The terminal end of the axon is branched and vesicular and is known as telodendria. Some neurons are covered by a layer of sphingomyelin, a phospholipid known as myelin sheath or medulla. Myelin sheath is covered by thin cell membrane, which is known as neurolemma or Schwann cell. myelin sheath acts as an insulator and prevents leakage of ions. So whenever an information is going from here to here, the myelin sheath prevents the information from leaking out of the axon. The Schwann cells that are present on the axon are responsible for maintenance of the myelin sheath as well. If the myelin if in where myelinated neuron should be present, if there is a non-myelinated neuron present, that is going to affect the speed of information transfer. According to the number of processes, neurons can be unipolar, which implies that there is a single process arising from the cyton, which is very common in the embryo, because there is not a lot of information transfer going on in that stage. Not a lot of complex, there is a lot of information transfer going on, but not a lot of complex information which needs to be very fast going on during the embryogenesis. Sometimes this single process is further divided into two processes. One of these acts as an axon, where the other acts as a new dendrite. These kinds of neurons are known as pseudo-unipolar. With examples are dorsal root ganglia of spinal cord and granule cells of olfactory bulbs. Next is the bipolar neuron, which has two distinct processes arising from the cyton, axon at one end and dendrite at the other. So there will be the cell body, there will be the dendrite, and there will be a very long axon. These type of neurons are found in retina, olfactory epithelium, vestibular and cochlear ganglia. Multipolar neurons have one axon but many dendrons. So they will look something like this. This is a multipolar axon. There are multiple dendrons going out of the cell body, but there is a single axon. Motor neurons are interneuron and multipolar in nature. Neurons of hydra and amacrine cell of retina have no definite cell processes. These neurons are known as apolar neurons, so they just have a cell body. Functionally, neurons, so this is based on the structure. Functionally, they are of three types: afferent or sensory neurons, which conduct information from receptors to the central nervous system. So for any information to be transferred, there is a receptor which gets the stimulus, which will then transfer the information to the CNS. The CNS then interprets this information and then sends an information to the effector, which can be a muscle, which can be eyes, etc. Nerve conduction from the central nervous system to the effector organs is a function is done by efferent or motor neurons. Interneurons or association neurons lie between motor and sensory neurons and are mostly present within the central nervous system. The nerve itself has two properties: excitability and conductivity. So when there is a stimulus applied, the nerves have the ability to enter a state of excitation and the ability to transmit that excitation in a particular direction. Both of these are very important properties of the nervous tissue. Just one moment. So the nervous system, even in its resting state, has a membrane potential to it, which is approximately -65 millivolts. The cell membrane of the nerve cell is said to be polarized when the negative potential exists more inside the cell with respect to outside. So there is more negative potential than outside. The inside has potassium, is high in potassium ions, and the outside is high in sodium ions. Both potassium and sodium won't exist simultaneously inside the, they can exist, but there is a gradient to how sodium and potassium ions are present across a membrane, across a neural membrane. The potential difference across the cell membrane at rest is known as the resting membrane potential, is approximately minus 65 volts. This resting membrane potential is maintained by active transport of ions against their electrochemical gradient by sodium potassium pump, and also by passive diffusion of ions. For active transport, there are carrier proteins located in the cell surface membrane. They are driven by energy supplied by ATP and coupled by removal of 3 sodium ions from the axon with the help of uptake of 2 potassium ions. So there is a net negative charge going on because there will be exchange of chloride ions as well. The passive diffusion of ions opposes the active movement of ions. There are certain sodium potassium pumps present on the membrane that facilitate this process. The rate of diffusion depends on the permeability of the axon membrane for the ions. Potassium ions have more permeability than sodium ions. Therefore, loss of potassium ions is more than the gain of sodium ions. This leads to a net loss of potassium ions from the axon and generation of a negative charge within the membrane. Then, when there is a impulse or a spike, there is depolarization. That implies that the the potential of the axon is increasing. The nerve impulse is known as active potential generated by change in sodium ion channel. These are voltage gated channels. At resting stage, these channels remain closed due to binding of calcium ions. When there is an action potential by sudden opening of sodium gates, the permeability of the membrane will change. The permeability of the membrane for sodium increases, because of which it enters inside by diffusion. This increase in positive ions inside the axon drops the negative potential inside axon, and there is a change of minus 10 millivolts in the potential difference from the resting membrane potential, known as the spike potential. Once there is a spike potential, this triggers a rapid influx of sodium ions, which leads to generation of the final action potential. This action potential can then travel throughout the axon of the neuron. First, the negative resting potential is cancelled out. At this point, the membrane is completely depolarized and the potential difference developed across the membrane is around 30 millivolts. It corresponds to the maximum concentration of sodium inside the axon. So now the sodium that was present outside is inside. The potassium goes out if there is a stimulus applied. So this the resting axon is now depolarized. Resting axon which was polarized is now depolarized. When the stimulus is over, then when the stimulus is over, the ionic balance is then reset and the axon gets repolarized. A fraction of second after sodium gates open, depolarization of membrane causes opening of potassium gates. Therefore, potassium is diffused out of the axon. This causes less positive charge inside with respect to outside, and due to repolarization, the potential changes from 30 millivolts to back to minus 65 millivolts. The neuron is now prepared for receiving another stimulus to conduct as described before. Now it is necessary to restore the normal resting potential by expelling sodium ions and taking potassium ions inside. Time taken by the restoration is known as refractory period, because during this period the membrane cannot receive an impulse. So what you need to realize is this is a single membrane where all of this is happening. There will be areas in where the stimulus is applied. Once the stimulus is applied, the membrane gets depolarized. It goes to 30 millivolts. The polarized area where the stimulus hasn't reached it is still at minus 65 millivolts. The information transfer essentially happens because the depolarized area then comes here to a polarized area. This depolarized area can then repolarized for the next stimulus. If the area is still depolarized, there will be no even if stimulus is applied, even if there is an impulse, there is no action from that impulse. Okay, so the type of signal that comes from the impulse can also determine what kind of action potential, what kind of potential is generated inside the next neuron. If it's an excitatory signal, then the action potential is going to the potential of the membrane is going to increase, of the axon, of the cell body is going to increase. If it's an inhibitory, then it's further going to decrease the action potential to a even lower value. So if the signal is excitatory, it will result in an excited neuron, whereas if the signal is inhibitory, which it will result in an inhibited neuron. Now this signal depends on what kind of neurotransmitters are present inside the previous neuron. If it's if the neurotransmitter is something like GABA, if it's something like glutamate or glutamate, then it will result in an excited neuron. If it's something like a GABA molecule, then it will result in an inhibited neuron. Okay, so in the place where information is transferred is known as the synapse. This is the area of functional contact between two neurons for transmission of information, which is known as the synapse. In the synapse, the membrane of telodendria, known as the presynaptic membrane, and the membrane of dendron of the postsynaptic postsynaptic neuron or the postsynaptic membrane. Between this, there is a space called a synaptic cleft. So the presynaptic terminal will release the neurotransmitter into the synaptic cleft. The postsynaptic membrane will have receptors for these neurotransmitters. It will bind once the neurotransmitters have bound to the postsynaptic membrane, it will signal downstream processing in case of downstream signals in case of the postsynaptic neuron. When action potential develops in presynaptic membrane, it becomes permeable for calcium ions, and calcium enters the presynaptic membrane. When vesicles burst by stimulation of calcium ions, it releases acetylcholine. Acetylcholine is one such neurotransmitter. The acetylcholine reaches the postsynaptic membrane via synaptic cleft, binds with the receptors which develop excitatory postsynaptic potential. So, we now know what are the different types of nervous system, how are the nerve structure, how neurons function. At this stage, are there any questions by anyone? Please let me know if there are any questions. Otherwise, I'll the move to some of the questions that I have. Okay. Let me just share the screen. Okay. So, the first question is, during early embryonic development, the cells that eventually form the nervous system, including brain, spinal cord, differentiate from a specific germ layer: endoderm, ectoderm, mesoderm, blastoderm. Does anyone want to answer? Yes. The answer is ectoderm. The nervous system division that carries signal from the CNS to muscles and glands is peripheral nervous system, afferent division, efferent division, or reflexive division. Just 1 second. It's the efferent division. 1 second. Okay. It's the efferent division. Function of the autonomic nervous system is regulation of involuntary functions, control of voluntary actions, processing sensory information, or coordination of reflex actions. The answer is regulation of involuntary functions. If any, please answer if you know the answer. It's supposed to, you guys are supposed to answer these kind of questions. Part of the brain that acts as the control center of involuntary activities is cerebrum, cerebellum, medulla oblongata, or pons. The answer is medulla oblongata. This has control centers for respiration and peristalsis, etc. Meninges are the outermost layer of the spinal cord, triple layers of connective tissue that protect brain and spinal cord, specialized cells of the peripheral nervous system, or fluid-filled sacs around nerves. The answer is triple layers of connective tissue that protect the brain and spinal cord. A patient suffers from a head injury. The doctor explains that a specific fluid in the brain and spinal cord which acts as a cushion to protect these vital structures from physical shocks is leaking through the nose and ears. This specific fluid is amniotic fluid, synovial fluid, interstitial fluid, or cerebrospinal fluid. The answer is D, cerebrospinal fluid. Srikanth is riding a bicycle and navigating through a winding path while maintaining his balance and coordinating his movements. The brain The part of the brain that is responsible for these actions is cerebrum, cerebellum, hypothalamus, or brainstem. The answer is cerebellum. Types of nerves that are part of the peripheral nervous system are spinal nerves, cranial nerves, spinal and cranial nerves, autonomic nerves only, sensory neurons only, interneurons. The answer is spinal and cranial nerves. Type of measurement that the neurologist use to observe and record brain's activity during EEG test is brain structure using magnetic fields, blood flow to the brain, nerve impulse conduction, or electrical activity of brain. The answer is electrical activity of brain. Type of nerve involved in smell perception is olfactory nerve, optic nerve, auditory nerve, and vagus nerve. The answer is olfactory nerve. So olfactory nerve is responsible for smell perception, optic nerve is for vision, auditory nerve is for hearing. Is gustatory is responsible for taste. So the name would in some cases suggest what function the nerve is responsible for. After a large meal, you notice that your heart rate decreases and your digestive system becomes more active as it processes the food. The nerve that is responsible for regulating these changes in your body's internal function is olfactory nerve, vagus nerve, optic nerve, or auditory nerve. The answer is vagus nerve. The delta waves in an EEG are associated to deep sleep, stress response, meditation, alert mental state. The answer is deep sleep. There are other waves that are associated with all of the others that are mentioned here. A person with a disease like multiple sclerosis experiences slower reflexes and difficulty in motor coordination. The part of the nervous system that got damaged and contributed to lower transmission of nerve signals is myelin sheath, axon, dendrites, or neuroglia. The answer is myelin sheath. So if there is a problem with the myelin sheath, it's going to decrease the speed of the uh, transmission of the nerve signal, because the ions won't stay inside the axon, they might move out of the axon when the polarization of the neuron is occurring. When a neurotransmitter is released from one neuron to communicate with another, it crosses a small gap called synaptic clefts, nodes of Ranvier, axon terminals, or neuroglia. The answer is synaptic clefts. During a muscle contraction, a person's motor neuron releases a neurotransmitter to stimulate the muscle fibers. The neurotransmitter that is typically released at the neuromuscular junction to initiate muscle contraction is dopamine, glutamate, serotonin, or acetylcholine. The answer is acetylcholine. The others are also responsible for glutamate is a neurotransmitter responsible for conduction of information, but that has a specific function. That's it. Are there any questions? Please ask if there something wasn't clear, or if there are any questions from the text or from the questions that I discussed. Okay. If there are no questions, then this is the end of this session. Thank you for attending.